Go.
Welcome everyone. Thank you for joining this live roundtable. I'm excited that we are able to do this. I'm Evelien Nijssen. I'm leading the marketing team at CellGenix. We will introduce the other panelists at a later time point. So today I would like to discuss where the industry is at for automating raw material processes in cell and gene therapy manufacturing. So what are the opportunities and the biggest challenges to overcome? So we already set up a survey prior to this roundtable to gather some insights from the industry. So for everyone who filled it out, thank you very much. If you haven't filled it out yet, you can click on the download content session button in this live roundtable. The survey will be available during the entire event and I think also a few days afterwards. We will publish the results after the event, in which we'll also include some of the discussion topics from today. For the ones who are joining us and watching us, please feel free to ask any questions via the chat, so I'm able to read those as well, so we can maybe incorporate them into the discussion. If you have any questions and we don't have enough time to answer them, please connect with me or one of my team members via the platform, so we can either have a chat on the platform or set up a meeting. Okay, so let me please introduce everyone. So we can start with Cloud.
Yeah, good afternoon. I'm here from Switzerland. My name is Claude Hollenstein and I am GMP manufacturing manager at Cutis AG.
Thank you, Claude. David Kunten.
Yes, hi everyone. David Kunten from the University of York. I'm currently entrepreneurial lead at an Innovate UK funded project to commercialize some cell-derived therapies.
Thank you. Brian?
Brian Poltolov with Broad Oak Capital Partners, where we're focused on investing in platform tools, technologies, and services to drive new therapies.
Thank you. Deanne? Let me see if she's online. Then let's go to Julia.
Good afternoon, I'm Julia Nemish. I'm a supply chain manager at GSK. I work in cell and gene therapies and I look after materials management.
Great, thank you, Julia. Kathy?
My name is Kathy Schneider. I work for Irby Biosystems, which is a new tools provider for microfluidic bioreactors, and I lead in the product management function.
Thank you. Sean?
Sean, you're on mute. From Sexton Biotechnologies, our tools provider, we have raw material reagent as well as processing and handling tools.
Thank you, Sean. Tom?
Hi everyone, good morning, good afternoon. This is Tom Heathman. I'm the Vice President of Commercial Operations for Ori Biotech. And we're developing a novel piece of manufacturing technology for the cell therapy industry.
Thank you. And David Lewandowski?
Hi, I'm Dave Lewandowski. I work with Brooks Life Sciences. We're providing cold chain solutions, process control, improving product integrity. Thank you.
Thank you. Kurt?
Hi, good afternoon, everyone. My name is Kurt Gillen from MedAce, based in the southern Netherlands, where what we call a biomedical workspace, where we support early stage cell therapy and medical device startups, especially in setting up their manufacturing.
David Huddle.
Hi, everyone. I'm David Hodel. I'm the head of business development for Invitech Cell Therapy Group. and we provide automated systems for cell therapy manufacturing and peripheral systems.
Thank you. Then we have Mark.
Hello.
I think Mark is having some technical issues.
Oh, yeah. Hello, sorry. It's Mark. I'm a senior scientist at the Cell and Gene Therapy Catapult. We work on industrialization of a whole host of products, from viral vectors to T-cell therapies to stem cell therapies.
Thank you. Diane, are you now there or?
Yeah, sorry, I was also before, but you couldn't hear me. Yeah.
Okay.
So hi everyone, I'm Diane. Currently from the music project, clinical project manager and coordinator and CEO of Move On Therapeutics in Foundation.
Thank you, Diane.
Thanks a lot.
Did we have everyone? I think so. So welcome. I'm excited to start the discussion with you. Thank you all for joining. So as I said, one of the topics I would like to discuss is how we can automate raw material processes in cell and gene therapy manufacturing. Since there is already a lot of effort in how to automate processes, and then since there's no industry standard for us as a raw material supplier, it's one of the challenges is really to see how we can offer solutions that allow you to incorporate or integrate our raw materials or from other raw material suppliers into these different systems, and there's no one system available. We also asked in our survey which raw material characteristics would enable to do that actually. And the majority answered closed system connectivity, which I think is not a surprise to anyone. There's a lot of effort in that and also a lot of discussions on how to improve that. Still some challenges. Other answers were global standardization, so that came up a lot, and also pre-mixed ready-to-use reagents. So we see a trend there as well. So to start with the first, so closed system connectivity. What do you think are the biggest challenges regarding setting up closed system connectivity between these GMP raw materials and closed automated cell processing systems, and how can we improve that? So what are the biggest challenges and what is still currently missing? Maybe Tom, would you like to start with that?
Yeah, sure. Yeah, maybe we can, first we'll start by getting a bit deeper into the challenge statement for some of this, because, I think there's a number of areas that represent a challenge for automation of raw materials inputs. First of all, closing those is a challenge that must be overcome. And really, when you're taking materials into the manufacturing space, the current state today is the materials are typically received in different formats. Mm-hmm. Some in vials, some are bags, some are bottles. And so that means that there's no standardization of materials coming in, and the manufacturers have to deal with that into the manufacturing process. Number two is the technologies. You're trying to insert those reagents, obviously in a clean room manufacturing setting, into different technologies. Many of those have different inputs and outputs as well, mm-hmm. which raises a second challenge. And then third is the manufacturing processes themselves that, there's lots of different products out there. But even if you take, let's say, a CAR-T manufacturing process, for all the various processes that are out there, there's slightly different ways in which reagents are used, how they're added, when are they added, how much you add. And that's typically based on cell counts or growth rates through manufacturing. So there's a number of challenges in there with that, but certainly, as you said, step one is closed systems. If you can do additions without requiring biological safety cabinets and take the risk of open manipulations at step one, that just kind of frames out some of the challenges that we see.
Does anyone want to comment on this?
Sure. So I think Tom hit on, I think one of the major points, but also, when we think about the GMP manufacturing, the supply chain that's feeding that, you're also then talking about all those suppliers that now are needing to think about, okay, how am I packaging, my raw material to get that to, my customer, that my GMP manufacturer, and what kind of information, what kind of data am I supplying them, on that barcode or on that RFID chip or whatever's in that, packaging material to feed into their system. So it's thinking about, from the supplier's perspective as well. What do we as, say, automation suppliers need to be able to set those, those stakeholders up to be successful?
And do you, David, so is that the first thing you discuss also with your customers, your clients, so what their requirements are and their expectations? Or do you rather have a standard for yourself?
Right. So that right now, I mean, as I think everybody has mentioned in our discussions prior to the start of this, that there aren't any standards, but there are sort of those broad buckets of, I need to bring in my raw materials in bags that I can sterile weld, or that I have, sterile connections, and then going back to those suppliers and saying, this is what your clients are wanting. This is what your customers are wanting, and are you capable of setting that up, or how do you close that gap?
Mm-hmm. Anyone else has also this type of experience, and how do you approach that issue?
Yeah, if I can maybe add to that. So as I mentioned, we work with early stage therapeutic developers. And what I see is that they have so many challenges simply from the product and how they're trying to get it to the patient, that all of these issues are an extreme burden to them. And I'm kind of throwing maybe something up here for discussion. And maybe Evelien or some of your colleagues supplying raw materials can pick it up. But I think it's also a question about who's responsible for setting these standards. And to me, that's part of a maturing industry as the ATMP field. In the past, all of this was coming from the therapeutic developers who kind of defined what they needed. But I think we're now getting to a stage where much more service and solution providers are out there who need to take up their responsibility. I'm not using this in a way of blaming, but it's more part of the stage of the industry, where I think, okay, what can companies like Sexton, for example, can do from a product development perspective? Can they include the question about standardization and what standards do we need as part of their future product development plans?
Yeah, great.
I would totally agree with that, with as it relates to the building of standards, but I do think it's a combination of responsibility. For sure, I think the tools providers, as you said, they provide platforms, and they're good. The regulatory side is also a really important aspect, because we certainly need a global convergence from some of the lead geographic institutional bodies that are setting those requirements and facilitating the transfer of information from a regulatory filing perspective. And so some of these public-private partnerships that are coming together and that are emerging, I think have an opportunity to play a critical role to set what is the need, what is the requirement, the expectation around componentry, around documentation, and then around the technical feasibility and format, such that we can really have a closed system that technically works, it scales to the right levels, it is compatible with the right types of materials, and ultimately it will support a global harmonized standard for the industry.
Yeah, that's actually a really great point, Brian. I think that's one of the issues. At least we see also that there are quite some initiatives already globally to set this up, to improve this. Sean, you I think you also have a bit of a regulatory background experience. Have you heard about some of the initiatives, such as the ISO technical standard? Could you comment on that?
Yeah, sure. So, from our position in the United States, there has been some legislation even that said we need to help support standardization on cell and gene therapy and regenerative medicine products globally. People are trying to work on that. But I think if anybody has had the the opportunity or the challenge of working on standard setting bodies, it's a very long and torturous process. And a lot of people end up bringing their own personal biases to that conversation. And that's why it takes so long, is because to get to consensus, it's a real challenge. And I think, Kurt's comment there about industry taking some responsibility on that. And what we've seen is that we go out and ask everybody what they need. And it really is the situation of you ask 10 people, you get 12 different answers back. Mm-hmm. And so maybe it is time now for industry to sort of step back and say, look, if we really want to accelerate standardization on these things, what do we think the most feasible way of doing that is? And then take that back to the therapeutic developers and the regulators and say, here's a proposal. Because it feels to me like everybody has sort of been talking about standardization, but no one's exactly sure what direction to go.
And if I can comment on that, Sean, I think you're absolutely right. I've read a great book years ago called Crossing the Chasm, where it went from early adopters to the early majority. And that always stuck with me. And this was about marketing, but I think this applies here as well. It does mean, though, that you will have to make choices because you can't do everything. But if I look at your companies and the company that Evelien is working for, if you look at your product portfolio and say, okay, we're together with 1 or 2 or 3 of our competitors, we are the market leaders. So let's get together to define what's important for our product portfolio. I think we can make big strides because I totally agree with you. Setting standards is a lengthy process. And although I also agree with Brian, leaving it just to the regulatory authorities is going to take decades, I'm afraid. And I work and I like startups, so I'm always looking at ways of how can we accelerate these things.
Thank you.
I think there's, so if I can just add to that as well, I think there's scope there for, way upstream in the development process for academics to get involved in this discussion as well. A lot of innovation comes out of academia, and progressing the science for a lot of these emerging therapies is something that's really going to set standards as well. And I think also cross-disciplinary interaction is really important here, and that academics, for example, start collaborating with industry in order to find out what industry is looking for, and also for industry to find out what academics are all about. I mean, obviously they know, but for that interaction to become a little more productive, I think a lot more collaboration needs to happen.
If I could highlight one great example that helped in this situation, standards are really difficult because they're going to cross so many different disciplines. An international society that I've worked with in biobanking created a document that was just a simple best practices. This was a consensus document, but it gives people great guidance on how to manage, certain collections and do certain things. And when I think about standards in cell therapy, I think about some certain buckets that, maybe we could break it down by these big topic areas, maybe apheresis, bioprocessing, data. There seems to be some big buckets, and if we could get some consensus around best practices as a starting point, it might, then be able to piece those best practices together to create some kind of a standard longer term. Mm-hmm. But it's really difficult in an industry where it's such a dynamic industry and there's so many variables and inputs that, to create a standard, how do you do that, right? Because we're still learning. So if we can decide on some basic guidance that says, hey, you know what? This is the size of stuff we should use or whatever, and just give people some boundaries to play within. That'll help the David's at the University of York's and and others that are in the early phase R&D really be able to set themselves up for success, right? So.
Yes, thank you. I think so. Go ahead. Sorry, Kathy, I think you wanted to comment.
I think, David, I kind of agree, but I think you're right. We have to figure out what's that first step. You know, is it a process? Is it something as simple as tubing, right? What is there a standard on tubing? Is there a standard on connections? I think the interesting piece with cell therapy is, you know, it's a convergence of different disciplines. You've got kind of more from the academic side, blood banking side, you know, that side. Then you have the bioprocess side, which has its own set of standards. Blood banking has its own set of standards. And cell therapy is kind of square in the middle of this. Like, so it's hard to figure out which standard to pick. Because when I go into commercial manufacturing, I'm probably going to want to adopt more of what's probably being done in bioprocess, because some of those processes, or at least well known, those materials are fully tested for commercial grade stuff. But then in the academic side, you also want to have that flexibility in the experimentation and kind of figuring out what that is. So I think we somehow have to figure out, I think you're right, it might not be standards, but it might be more guidelines of, we need to accept the following principles as guidelines going forward.
Mm-hmm.
Yeah, and oftentimes in the early development of these technologies, there's a lack of that know-how and that expertise, where when you start translating or scaling up, you know, the problem becomes, where do I even begin? And, you know, is this even scalable? And I think it needs to start very early on, having that access.
Yeah, we have also experienced that now we have our manual process and we are already in phase 1. And now thinking for scaling up and putting automation into the game. And then I just ask, okay, what is enough for you, for the authorities, for design freeze, so that we can compare the different steps which we automate, that we can prove that it's the same thing, that we don't have to go back and repeat everything. And there is again no answer. Yeah, I have to think of ways that this would be enough for me, but maybe in 6 months someone else comes and says, no, it's not enough, you have to go back to the bench. And it's really difficult, because these decisions you make for planning 5 years in advance, and everything is very complex, and there is really no like guidance where you can follow.
Yeah, I think, Deanna, you make a really good point there when you know, the other variable we have to think about is scalability. You know, what does GMP manufacturing and the supply to those GMP manufacturers look like at, you know, 500 patients a year or 1,000 patients a year versus 5,000, 10,000, you know, autologous versus an allogeneic process. And, you know, when we're working with manufacturers, you know, their mindset is always, we solve one problem at a time. You know, let's solve problem A before we move on to problem B, because if we try to solve them both, we might break the system and have to start over again. So it's a challenge to think of, you know, through that whole range of scale, and, you know, how do you provide for, you know, the ability to keep your manufacturing system closed with everything that's going into that process.
Yeah. Exactly. So that's a good point, David. I also want to bring it up because I think there's also quite some difference between autologous and allogenic. So scale up versus scale out. And for us as a raw material supplier, it's even, you know, if you provide media in 1 liter or in 5 or 20 or even 50 liters, that changes everything. So then also standards, but also packaging, connectivity. So at every different level, you have different challenges.
I do think that there are things we can do today, though, right? I mean, you know, as was mentioned earlier, developing these standards, putting all that together is going to take a very long time. And, you know, as the industry is in flux and things are changing, it's going to be tough to nail that down. But, you know, today, I think, you know, there are approaches. For example, you know, Ori is a manufacturing tools developer. And what we're looking to do is integrate further upstream and say, okay, if we're developing a piece of equipment that's going to need to have additions, things like culture media, virus, cytokines, all these types of things, what can we do to work with some of these suppliers so that once this is integrated into a client's process, can we offer them a solution to have their culture media or their reagents, whatever they need, in a consumable, in a format that we know is going to automatically sterile connect to our device, rather than say, okay, we're supplying a piece of manufacturing technology, you have to figure out how to get things in and out of it. And so, I think today there should be a much bigger onus on the tools providers to take a step and say, okay, I'm going to take some responsibility here for integrating further upstream in my supply to have a solution for a customer that's not just, well, put it on a bench and manufacture a product. And, we can do that today. That is something we could do. We can each talk to partners upstream, downstream, and start to integrate some of this. And, that way it at least clears a path through, if you have a technology for phase one, where you can have manual open additions, 10 products, 20 products a year, you can manage that. But when you get to 1,000, as David Hollow says, it just doesn't work. So, I think if the tools providers can take a step in doing that and enabling scale by working within their supply chain, I think that's one way that, we could actually take some steps here.
I think what's really important there, though, is that we need to think about how we're trying to integrate and kind of, if you will, at what level or what dimension. Because if you think about this, I think the traditional method is to take a step of a workflow process and try to focus on that step of workflow. And that's where a lot of the tools manufactured traditionally have focused. But I think the point that was brought up earlier around allo versus auto as an example, and then your comment about Ori and the approach around integrating upstream and downstream. You're not going to be able to solve every problem for every therapy for all of these different formats, but to take a classification or a category and then to be able to integrate up and downstream for at least a set of autologous processes that are geared for a certain volume of ranges and scales. Then I think you can actually start to get a sizable bite that can get a seamless workflow and get a set of tools, instruments, reagents with the right level of compatibility and physical connections, virtual connections, data connections that's actually going to work seamlessly and integrate across. But I think that's a little bit of a different spin than maybe the industry has traditionally taken, where it just focused on a specific workflow process and looking at for scalability at all levels, at all ranges. And so I think that's a little bit of the different lens we might need for the CGT space versus traditional biologics or small molecule.
But I think Tom's point is quite pertinent in terms of getting that harmonisation between the tool makers and suppliers when it comes to connectivity and stuff like that, and the tubing sets, etc., would really help bring that standardisation.
I think, Tom, to your point, and there are already some of the tool suppliers that are starting to act in that way in terms of thinking about up and downstream and how do we interact. And one of the goals that we have as a vision is to try to think about that integration across things instead of necessarily developing out proprietary unit operation systems, figuring out ways that we can help people do that closed system transfer from the raw material bags, no matter what the source is, into an out bag, no matter what output bag they want or what the source is. And I think that, we're working hard at trying to develop the relationships with other suppliers so that we can talk to our customers and say, here's some additional ideas and here's some other customers that might be able to fit with your needs on that and interact that way. I think there are other groups that are approaching it kind of maybe what Brian was alluding to, where it's, we're going to exclusively work with these other customers and we're going to set up a very defined process. And, I think the flexible automation concept has been really gaining a lot of steam. And I think just talking to your suppliers about what you need and with who you might be working could be really helpful. You get a lot broader interaction.
Well, and to Sean, to your good point, and and I actually want to make 2 points really quickly. 2 years ago, in the Phacilitate Special Interest Group, one of the groups, their sole focus, for those couple days was talking about standards, in particular with, not only, connectivity, but also with single-use systems. So it would be probably if there's a way to access that pretty quickly for, our audience, it'd probably be, a great recommendation for them to take a look at. The second challenge, I think, is, when we think about the suppliers in this equation, how do we work with them? And I say we being some of the suppliers of automation, how do we work with them to get them set up to be able to package their reagents, package their products in a way that can be easily adopted? Because our manufacturing partners or our manufacturing customers, they're not going to slow down, and they're pushing on us to say, hey, we need our raw materials packaged in a way that we can use them in our system. We don't need it a year from now. We need it a month from now. So I think one of the things that is incumbent on us is to be able to work with work with those suppliers to say, we can help you get to that position where you can supply all these manufacturers with your raw material. It then becomes a question for those suppliers as, is it worth it? Because there is going to be some capital investment involved, and you've got to do that math. Does that investment pay off in the mid and long term?
Yeah, that's a good point, David. That's why we also at CellGenix organize these open discussions between all these stakeholders. And we have this cooperation also with Ori Biotech. Like Tom said, that their approach is really to work also with different raw material suppliers, because sole supplier dependence is also risky. So I agree also we have to go in that direction. Kurt, do you want to add something?
Well, I actually want to re-emphasize something that came about earlier in the conversation. When Deanne said, "Okay, we're phase one now. We need to scale up," and Tom actually already gave the answer of that problem is maybe to be pragmatic and say we're not going to be able to solve everything fast, but can we already start thinking about this is phase one, this is phase two, kind of coming back to what Brian said. Maybe make a choice about number of production units that we need to be able to produce. Whereas a company like the one from the end can say, okay, here's my solution for phase one. And I know in phase two, I'll have to change my manufacturing process. But at least there's already some comparability between the different setups. I think that would be a really good approach to discuss and say, as solution providers coming together, can we think in those concepts where a company like the ENDS and all the other therapeutic developers can choose between the different systems where there's already comparability done from the providers, but knowing that, in phase one, she can't afford the full blown automated system. That might be a pragmatic approach, I think, to at least make some steps forward.
I think comparability is an interesting one, because when it comes to closed or automated systems, then cell and gene therapies, the complexity is such that small changes in your system may not behave the same as small changes in someone else's cell system, and it does end up being on the client's end a matter of optimization. No matter how well characterized the process is, I think it's really difficult to find out about comparability before implementing the system. That's our experience anyway, but it's definitely something that's...
David, as much as I agree that that's the experience today, I would kind of challenge that, and that needs to become something of the past. Because to me, that is, and I'm an engineer background, it's a lack of engineering of the process, and that comes down to in process analytics and what have I. I live in the southern Netherlands, where Lonza has their center of excellence for cell and gene therapy. My previous company used to be building clean rooms for them. It was a great business because they're growing tremendously. But I can tell you, economically, it's not a scalable model. So we need to be moving away from the operator knowledge and the Fingerspitzgefühl, as they say in German, to a real, very well designed and engineered process where this becomes a thing of the past. But nevertheless, I agree that that's what we see today.
In terms of automation, right, you can break that down because, clearly if you're automating, let's say, a culture step or a wash formulation step, there's going to be a high comparability risk in that. But, if we're really talking about automation of addition of reagents or taking of samples, closing, automating that, the risk may not be as high to the product. And I do think, when seeing a manufacturing process, a number of manual additions and manual sampling, if you could just remove that from the process, that would be a giant leap forward, going from 10 products to 1,000, right? You'd probably take out 50, 100 people from your process because it's so frequent. So, I think there are chunks that we can take out. As I completely agree, David, that, automating the entire process, a number of people have tried it and it's incredibly challenging for a number of reasons. But I do think there are some smaller steps, particularly in the spirit of this conversation, with additions or subtractions from the process that may not be as high of a risk to the product. I'm talking to the product. So yeah, there are steps I think we can take just for automation.
I think, Kurt, you pointed out like showing a map, right? If this is step one, this is phase one, this is phase two. I think one thing, at least when I've sat through these conversations before, we seem to forget that there's customers in phase today, right? So if we come out with kind of what we would say is our recommendation from from an industry perspective, we have to provide that map of, also considering when do you phase this in? If you're already in a phase one, if you're in a phase two, do you consider this now? Or, what is the grandfathering effect, for the lack of a better term, as you can continue through that process, right? So I think standards still are recommendations, as David said, is probably still the best first step. But then I think as part of that group, and I think it's a consortium of folks that gets together, like, what Bioforums had and things like that, that kind of makes that recommendation, but also the map of, okay, and here's how we thought of the whole problem.
Yeah, and if I can answer that, Katie, I think what Brian said in the beginning of the conversation, the public-private partnership needs to play a role in here. Again, referring to Lonza, they're scaling up the process of their clients, and the only thing that they want and their clients want is to get to the patient as fast as possible. In the way that they're doing it currently, there is no direct need for either party to make investment in improvement of the processes. But I think this is where public money or early stage academic groups, as David mentioned out, can play a role and say, okay, we have different interests to achieve, and they can definitely play a role that will then make the entire industry move forward. But you're absolutely right. If you're in process now today, it's a huge investment in making any changes to what you're doing today.
Just a thought. So I think everyone's focusing on their process. Is there also exchange between experiences from cell and gene therapy manufacturers? Because I think we can also learn a lot from each other. So not just, I think it's good to have a conversation between the raw material suppliers, then of course the ones that are manufacturing the therapies, and then ones that are manufacturing the devices. But I think also the cell and gene therapy manufacturers themselves that are developing it. Do you think that's already existing, or that's something also that you think should be good if we set that up? Brian?
They play a critical role in this, right? It's at all different levels, right? It's at the tools provider, it's the innovators, it's the regulators, it's the translational centers, and some of the academics. And I think that's the beauty of some of these partnerships and these collaborations, is they do bring together a number of vested interested parties, each of which have a critical perspective. And I really think the industry is still at a formative stage that I think we can really have that strong reach out, have that strong connection, and build a lot of those kind of those early connections that are going to set the standards. And so I do think we'll see a convergence. It doesn't happen without the pharmaceutical developers, for sure. There's no way. It actually requires all of them. And then what the public can do is really just helping to provide that forum, and so that way we can actually have thoughtful, fruitful discussions with the right entities, and hopefully one day soon in a physical, real, connected setting.
And I think to your question, Evelyn, those conversations are happening now, between the pharmaceutical companies and automation providers and the suppliers, some of their major suppliers, because they really have to happen. I mean, you're looking at the Novartises, the Kites, the Juno, CellGene, BMSs, the Bluebirds of the world that are they're either in commercial manufacturing now, they have a product on the market, or they're in very late stage and setting themselves up for commercial manufacturing. And, their viewpoint is very much of, we have to close our systems. We have to take that system that we took from an academic and scaled it to a point where we can get to clinical approval. Now we've got to automate that. And, the first consideration that they have is don't mess with the biology. Automate this, but don't mess with the biology. Oh, and now, well, we also have these peripheral systems, where we're using hundreds of liters of media a day. And, we have to be able to, get that into our manufacturing process, but in a closed format, so we keep everything closed. So the discussions are happening. It's actually taking that information and getting it in a forum where it disseminates to, the whole supply chain, and saying, this is where those pathfinders are pulling the supply chain. And I think the easiest thing that they've been able to do right now is tell us as suppliers, we need this in a closed, some sort of packaging format that we can use in our system very simply, whether it's with sterile connections, whether it's with tube welding, and then go from there, solve the next problem and the next problem. And we'll get to standards, and we'll get to standards that everyone really sort of accepts. But that's a longer term proposition right now.
Yeah, and if I can add two things to that, I mean, I've been throwing out the question about other people taking up their responsibility, but that's actually something that's built into the business model of MedAce, where we offer a work learning environment, and not about closed systems, but really about clean rooms and quality management system. And sure, our clients pay a fee for our services, but they get all the data and all the knowledge and know-how that they need to know. And any changes in the regulatory in Europe, we take care and we update them. So it's a new model that we're building. One other party that I do want to mention is the Phacilitate group as well. And David already mentioned the automation discussion that takes place once a year, where really people come together to discuss certain topics. That has been a phenomenal meeting in the past with some great insights. And I think some really good discussions that started the collaboration. So I think, everybody can play a role in this if they look at what they're doing and how they're doing things.
Yeah, definitely. Claude, do you want to add something?
Yeah, very interesting discussion so far. I mean, coming from I would say, relatively small therapy manufacturer that is in a rather late stage, still doing mostly manual, but we started automating relatively early, and we have a few processes that we were seeing for different modules that are available on the market, and we're testing them in an R&D environment, and already there we kind of see that every equipment, especially those that try to be multi-purpose, they have their own set of connectivities, and some of them can do welding, others cannot. So how do we deal with that at the same time. We're building our own equipment, or we're planning to build. So which one should we go? And another problem is, we're dealing with suppliers that are relatively small as well, that have not yet invested anything in automation or in closing systems, or connecting to closed systems, because they mostly used to produce small scale. So it just feels often that it comes back to us when we're not well advised. Especially in the beginning, it can be quite overwhelming when you don't know the field very well, because we're just basically out of university. And I mean, it's good that Kurt said, MEDES is supporting the people there with the knowledge, with the quality systems already in place, with the GMP system in place. I mean, we are also building a GMP. So I just feel like in the end, we want to have our suppliers supply us their media potentially in bags. And then in the end, it's just another project on our shoulders, other than, okay, well, what could you offer? What kind of connectivity? What kind of bags do you offer? And how is it validated? Do we have to validate it? And it just often feels that it can be overwhelming, but I guess it goes back to the standardization that even a small bit there can already help companies like us to orient themselves.
So some support in setting up this process and making certain decisions would be very helpful. So regulatory, but also just how to improve your system and how to make sure that you meet all the automating requirements. That's interesting.
But I think on that, there is a lot of help out there in terms of, for example, the Cell and Gene Therapy Catapult. That's why we were set up. We've got a lot of experience in helping customers automate their processes and stuff.
Yeah, sure.
I think it's worth. Sorry.
Yeah, I think
it's worth.
Oh, sorry. I think we're kind of a bit mixed up. Claude, did you want to?
No, I just wanted to add what you said before with the automation, Phacilitate automation roundtable. That has been really helpful to see who has the experience and who we can approach, or what are the general topics in the field.
So that helped you a lot. Yes. David, what did you want to say?
Yes, well, yeah, I just thought it's worth also thinking about, wherever you are with your development, having an appreciation of what your plans are further down the line, and integrating an awareness or a mindfulness of the prospect of automation can be really helpful. And so if an early stage sort of therapeutics developer innovator is, then sort of burdened with the thought of, well, I have to take my bench process to an automated state, that's the earlier you integrate that thinking of this is possibly going to be something I need to automate, then that's, the easier life is going to become. And I think that's a cultural change that needs to happen as well in the community. And something that, roundtables like this can be really helpful for.
As well, I think, David Hoddle kind of brought up the statement of, well, there's 1,000 challenges to face as you're moving a product through the development cycle. This is one, but there's probably others that are bigger. And, certainly looking for most products coming into an early phase, preclinical into clinical, even getting reagents that are of, for example, GMP grade, or of a grade that you would be happy to take through clinical trials is not trivial. There are certain reagents that can't be sourced in that format. And then, as has already been mentioned, single source suppliers for most critical or key proprietary reagents, are two huge challenges that, developers are probably have higher on their list, rather than how do I remove a bit of complexity from manufacturing? How do I close and automate my systems, remove a little bit of cogs? I would be more worried about the previous challenges than some of those. So there may be a maturity element here of, how do we move through and suppliers start to gear up for what is a more commercial industry versus, where we've been for a long time, which is more of a, early phase academic, and, somewhat being a victim of the success of the products, that these products have shown such high efficacy that there is a demand from all of us to get these to commercial and to patients, but there may be some maturity required on even the reagents themselves coming in, and my sense is that that's probably a priority over some of the things we're talking about, which might be some of the reasons why there's been limited conversations to date in this space.
Yes, so I think that's a good point, Tom. So also in our survey, we asked what are the biggest raw material challenges, and there are quite some mixed answers. So there seem to be different ones. And one that popped up was really that there is no GMP grade available for some raw materials. Also depends on sole suppliers, high cost. But we all know that manufacturing cell and gene therapies is quite costly. Well, closed system connectivity we already mentioned, and lot variability. So especially also with serum, we see that, but we also have some other raw materials. Also depends, of course, also on the quality that the supplier offers. So I agree, it's not just automating and how you're able to automate processes, but there's some other challenges that come into this picture as well that we should address. What do you think are the biggest challenges regarding raw materials?
That directed at me, sorry.
I don't know, or someone else, sorry.
Okay. Yeah, I think, as I said, those are a few that I've seen within manufacturing, right? Certainly single source suppliers is a huge problem because as a developer, it puts you in a very bad position, more so if your product's successful. We've seen that a number of times with products getting closer to commercialization, having to secure supply of key reagents and key formats for the product, and developer or the reagents providers turning around and asking for royalties, asking for milestone payments, success-based payments, all those types of things, which we've seen with most of these commercial products. And I know, we mentioned a few commercial providers, a number of them are going through that right now as they're launching products that they're having to negotiate and really have no choice, right? If you have a single supplier for a key reagent, then you're going to pay whatever they tell you to pay. And, I think that's one huge challenge that the industry has to overcome of, gouging developers and taking advantage of that, which, understood, if you have a proprietary reagent, it's, and you're the only one, that's fine. But until there's other options, there's not going to be really a competitive space there. That's going to lead to innovation and for people to change. And then, as I mentioned, just the baseline of, GMP versions, quality of these reagents, lot to lot, right? There's just a long way to go in terms of a stable supply of these reagents. Because, I can say from a manufacturing perspective, viral vector is a good example of a high lot to lot variation raw material that has huge implications in manufacturing. Changes fundamentally how much you add when you find out what the specifications are in a reagent, right? That's not really manufacturing in a traditional sense. So, I think there's some of those earlier challenges that really need to be ironed out. And there's no, finger pointing at all, right? It's just, as I said, it's a maturity thing that the scale's not been there to accommodate companies going building out huge facilities and high quality manufacturing of reagents and supplies, right? It's just not quite been there yet, but I think it's time to get a bit of competition in that space, to be honest, and for people to enable that, to actually get some innovation there and support the developers and not just take advantage of, very good positions in the market, let's say.
Can I throw out an example, which is actually outside the cell therapy space, but it kind of addresses to what Tom says. One of our clients is called Mozambique, and they don't make therapeutics, but they make hamburgers, lab-grown hamburgers, grown out of the cardiology department of the university, because in the end, it's just muscle and fat cells that you grow in large scales and, huge scales compared to therapeutic. And one of the biggest issues for them, obviously, is cost of goods. The first hamburger cost about half a billion, if I remember correctly. They're still up to the scale of like $50-$100 per burger. But what they did is they looked at the media and said, well, what's actually in there and can we change these? And they're doing a completely transformation of how they make their media. Now, obviously, standards are lower or different, I should say, than what we're looking at. But they are looking at something to really lower that cost of media by 100,000-fold. And this is outside of our space, but I think we should keep an eye on those kind of developments and say, can we use that innovation? Because I think Tom is absolutely right. You will see a lot of innovation in the industry driven out of all these problems. And how can we make that something where we can lower the cost of goods for our therapeutic developments as well? So I think Tom is right. The challenges are there, and it's up to the innovators within the industry or outside of the industry that can actually transform some of these or change or overcome some of these challenges.
But that can lead to a different sort of challenge in a sense where you're maybe looking at metabolomics to get a more customized media, which means you're losing that sort of standardization, which might actually increase your costs.
I think that's exactly right. And I spent a lot of time in the media development world. And for sure, there's a high level of cost in the development. And where you see value in why this problem's been solved in biologics and the like is because we've gotten to a place where it can support scale, it can support a long runway of commercial production and manufacturing. And I think there's a little bit of a trade-off there, as you said, in leveraging the infrastructure that exists right now, in taking advantage of that manufacturing scale, in getting the right level of collaboration and partnership between the innovators and the tools providers. I think that we need to get those conversations happening, and we need to be able to, as sooner versus later, get to scale and development, get to some level of confidence and standards, get to a place where the product will be regulatory acceptable in the US and in Germany and in Japan, et cetera. And then we can actually more rapidly get to a place where the cost, the manufacturing cost, the scale, the commitment, and the long-term sustainability of the supply chain will be there, and we can leverage what exists already for other compounds. Until we can do that, and until we can facilitate the collaboration and those partnerships and that long-term commitment to success and to continued innovative and collaborative development, we're going to struggle with this issue.
I think there's an important distinction in how you get to that scale issue, autologous versus allogeneic therapies too. I mean, if you're looking at media development and standardization on that, where in an allogeneic company, you can look at what that large, long, what you can realize commercially as a developer of a supply versus on an autologous therapy where you may be working on something for a customer that is going to treat 400 patients for the year. And it's a real challenge.
Yeah, I agree with that. And well, first thing, Kurt, you're never going to have me looking at a hamburger the same way again. And, to actually touch on Tom's point a little bit that he started with, I think the two major challenges that we're seeing right now from our clients that are in GMP manufacturing is, number one, the sole supplier. You're exactly right, Tom, there. But also having reagents come in and having raw materials coming in a closed system connectivity format. We're talking a lot about media, and to those suppliers of base media supplements, one of the critical challenges that we've been asked to solve with some of our clients is how do you automate a system where you're manufacturing 4 or 5 different media recipes with all these different components, and some of those components come in vials that you have to make down prior to, or they're being received in vials in a powdered format. Some are being received in larger 50-liter containers, some are in 10-liter containers. How do you do that and then aliquot that formulated media into packages that you can then use in your manufacturing process? And those are some of the challenges that our clients are working with, and they're asking us, hey, can you go back to our suppliers, work with them to help them get to not only those sort of standards right now, but how can you help them get to a position where they can package their product in such a way that we can receive it and get it into our process right away? You talk about reducing COGS. That's going to be a huge piece of reducing your COGS when you take that labor component out.
Yes, I think we're almost at the end of our session. I thought it was really interesting, so I hope we can continue it also a bit over these 2 days. Like everyone said, the most important thing to get this information, this movement going is collaborations. And setting up, I thought that was a good one, basic guidance and ranges and scales, and to see where we start. So take it in steps and not try to optimize everything at once. I hope that we will be able to have the automation meeting from Phacilitate soon in person again. I think that will be very helpful. I know that they already have 2 discussions, one today and tomorrow another one, also as part of this group. So I think it would be good to talk to them to how we can take what we discussed today and transform it into a useful meeting to work on that further. So thank you all for joining us, and thank you for all your input to the panelists, and have a great meeting.
Thank you. Thank you. Thanks. Bye-bye.
Thanks all.
Thank you.